Alright, so in this lecture, now that we've kind of finished an overview of cell anatomy, we're going to go back to cell membranes and talk about the real function of cell membranes. And ultimately, that function is going to be to delineate between two compartments and enable us to build up different concentration gradients in those different compartments. Now, before we go there, let's review kind of the structure of the cell membrane. Remember, the membrane is made up of different biomacromolecules. The base layer, kind of what forms it, is those lipids that form the lipid bilayer. Again, the hydrophobic tails are going to hide from the water on the inside of the membrane, and the hydrophilic heads are going to interact with the water on either side. This creates a barrier that's very robust, right? Chemically robust. Now, spice that up with some proteins. You've got to throw proteins in there because what do proteins do? They do stuff. Right? This membrane needs to be selective and selectively permeable. Right? So you have to know what things get to come across and what don't. What does that? The proteins. We're going to decorate the lipids and some of those proteins with carbohydrates. We have glycolipids and glycoproteins. Remember, we're going to combine those biomolecular classes to increase our diversity. So when we look at those proteins, we're going to see again that the regions that are interacting with the inner part of the membrane, that hydrophobic region in the middle, as you can see outlined here, those, in this case, these membrane-spanning domains, are going to be full of hydrophobic amino acids, right? So remember, going back to proteins to review that primary structure of proteins is the sequence of amino acids. And when I study that, I'm going to find regions that are very rich in hydrophobic amino acids, and that allows me to predict that maybe this protein is an integral membrane protein. Maybe that hydrophobicity is important to anchoring it in the membrane. Now, anchoring it in the membrane means that it's not going to float out and leave the cell or float into the cytoplasm. It's anchored in that vertical, or in this, the way it's oriented here, vertical up and down direction. But what about lateral movements? And so what I want to introduce here is what's called the fluid mosaic model of the membrane. Because in fact, this protein can move laterally or float in that thin layer of lipids side to side. Right? So the original experiments that looked at this, you would take one cell, in this case a mouse cell, that had membrane proteins, and you would decorate those membrane proteins with a dye, and that dye would be pink. So you could distinguish them from another cell's proteins. In this case, were a human cell, and those proteins you decorated with a purple dye. So you can force these two cells together, and that lipid will fuse and form one big cell. And so the question then is, how will those proteins move in the membrane? Will you get half a cell that's pink and half a cell that's purple, or will they intermix? And as you can see on the slide, that after an hour they were well mixed. And this supports this idea of a fluid mosaic, that you have this two-dimensional film of fat, the lipid bilayer, in which these proteins float and can move laterally. So remember, a mosaic would be like a picture. You build a mosaic, it's a picture of little pieces, of little chips of ceramic or what have you, and you build a picture. But if it's fluid, it means those pieces can move around in it. So this is what we think of when we think of kind of a model of the cell membrane. All right? Remember, it's got proteins in it because proteins do stuff, but those proteins can move around laterally. So when we want to cross that membrane, we talked about the ideas of proteins being important for that. Now, I want to introduce the idea of bulk transport across the membrane as well. So a special word called exocytosis, exiting the cell, allows us then to take like a secretory vesicle. Remember, a transport vesicle from the Golgi that's headed for the plasma membrane, that actually fuses the plasma membrane and release its contents outside the cell. Right? So we do this when we want to secrete a protein, antibodies, hormones, what have you. Exocytosis. You can do the reverse, endocytosis. There's a couple different examples of endocytosis that I'm not going to hold you responsible for, phagocytosis and pinocytosis. But endocytosis, entering the cell, into the cell. This is where the plasma membrane would kind of indent, fold back on itself, and form a vesicle from the plasma membrane, and then pinches off and comes into the cytoplasm. Right, so this is the kind of vacuole that could then interact with a lysosome that contained food from outside the cell to be broken down for the cell to use. Right, so crossing that all-important membrane that delineates the cell from its environment can happen by exocytosis or endocytosis, and we call this bulk transport because we're just nothing's actually moving across the lipid bilayer. Rather, the bilayer is manipulating the environment. It's either opening up to the outside of the cell or closing in and bringing something to the inside of the cell. Remember, those proteins, though, can form channels or carriers to move things across the membrane as well. So the next big question when it comes to biology is how and why do those things move across the membrane? How do we determine what needs to go across? And how do we move things across that are expensive to move across? And so we talk about diffusion, this idea of things moving down their concentration gradient, sometimes through the membrane, because the membrane is just a little bit leaky, sometimes through a pore. We call this passive if we're going from a high concentration to a low concentration. Or we could put energy into that movement and move things against their concentration gradient. We have a low concentration outside the cell, and we move things against that concentration inside the cell. That takes energy, right? This introduces the very, very important concept in biology of diffusion. And this comes back to physics as well. This comes back to the second law of thermodynamics. So thermodynamics is a component of physics that looks at heat and how heat moves, how energy moves through a system. So the second law of thermodynamics talks about order and disorder and puts numbers to those, that I can measure the amount of order in a system. And what the second law says is that the tendency of the universe is always to increase in disorder for spontaneous reactions. So if something kind of happens, if something can kind of happen on its own, it's increasing the disorder of the system. And diffusion is going to follow this law. So, for example, if I have a situation here on this slide, you can see the molecules of dye on the left in the upper left corner. And they're ordered. They're more packed up on one side, right? That's considered a higher state of order. And so what's going to happen is just what you would think would happen based on your experience. They would diffuse through this membrane that has holes in it until they're equal, until they have equilibrium, until they're moving back and forth across the membrane at equal rates, and you have the same amount of dye on both sides of the membrane. Think about if you took a bucket of paint, red paint, and dumped it into a swimming pool. Right, so that bucket of red paint is very, very deeply red. But you dump it into the pool, and then you're going to get red water right there as the dye is very heavy in concentration. But if you gave it time, days, perhaps weeks, then you would have a very lightly colored pool, a light pink pool, right? As all those dye molecules spread out, diffuse out from a region of high concentration to a region of low concentration. And that's going to be our definition of diffusion. This movement of solutes, remember something that's dissolved in solution, remember water is our solution, our solvent, right? And molecules are going to go from regions of high solute concentration to regions of low solute concentration. So things want to always move down their concentration gradient, and this is based on the laws of physics, specifically thermodynamics, the second law of thermodynamics, okay, that we're moving towards more disorder. So this is independent of other dyes in solution. So if you look at the bottom, if I have orange on one side and purple on the other, again, the orange is going to diffuse towards where the orange is not. The purple is going to diffuse towards where the purple is not until they reach the state we call equilibrium. We have equal concentrations. It's a really, really important concept. It's going to drive a lot of life. It's going to actually drive how we derive our energy, how we make ATP, but we'll talk about that a little bit later. Now, this is so cool because this aspect of the universe actually we can harness to do work. All right? So this is what you call an osmotic pump. So in this case, you've got two solutions on either side of a selectively permeable membrane. So this membrane is selectively permeable because it allows water to move across, but not the solute. So the purple dye in this case cannot move across the membrane, but the water can. So this is now a very special case of diffusion called osmosis. It's water going from regions of high water concentration to regions of lower water concentration. And so this is a little bit confusing because all of a sudden now we're talking about what has always been the background and kind of what things are dissolved in, but now we're actually talking about that water in the background as the thing that's moving around. So again, if you look at this picture on the left, you can see that you have two equal volumes of water. There's the same total volume of liquid there. But if there's more dye on the right than in the left, it means that there's less water on the right. And things want to go where they're not. So if water can move across the membrane but the dye can't, water is going to start to diffuse across that membrane until you have equal concentrations of dye on either side, equal concentrations of water. So again, this is called an osmotic pump because you can actually have that column of water to lift things. Right? It actually can do work as water moves from an area of lower water concentration to an area of higher water concentration. So this is such an important concept. Let's review it. So remember, diffusion is the movement of solutes from a region of high solute concentration to a region of low solute concentration. Osmosis is now going to be very specifically referring to water and water only. It's the movement of water from a region of high water concentration across a semipermeable membrane to a region of low water concentration. Now, in this definition of osmosis, we have to put in that semipermeable membrane, because if we don't, we'll just have regular diffusion, right? So it has to be a semipermeable membrane that only allows water to move across it. And the cell membranes do work that way to a certain extent. Because water is so prevalent, it can sneak across the membrane. In some cases, we have specific channels to allow the water to move across the membrane. And what that does is create a specific challenge for cells. And this is the challenge that we call tonicity. All right? So if things always want to go where they're not, and specifically water wants to move from where there's more water to where there's less water, we have to think about the solution that a cell is in. So if a cell, in this case the top row is all animal cells and the bottom row is all plant cells, the top cell in the middle is in what's called an isotonic solution. It means there's the same amount of salt in the water around the cell as there is in the cytoplasm. So water then moves into and out of the cell at the same rate, and the cell is fine. Now, in a plant cell, the only difference is you've got this plant cell wall. Now, this plant cell wall is like a box or outside the plasma membrane. And that's going to come into play if we go into a hypotonic solution. So now let's take that animal cell and put it in pure water. So pure water means there's nothing dissolved in it. And compared to the cytoplasm, which is full of all the things that we need to do life's biochemistry, salts and proteins and carbohydrates, all that kind of stuff, there clearly is less water. There's more concentration of solutes inside the cell. So those solutes want to move out of the cell, but they can't because the cell membrane's there. But water can cross semi-permeable membrane. So the water moves into the cell because it wants to go where it's not. There's less water, quote unquote, in the cell than outside the cell. Now for an animal cell, the water moves in faster than it can move out, and ultimately the cell will blow up like a balloon and burst. It lyses. This is why it's really important to maintain our internal environments, to keep all these concentrations just right, to avoid this tonicity problem. Now, a plant cell is a little bit more robust in this respect. I can pour pure water onto the roots of my plant and they don't burst. Why? Because of the cell wall. So in this case, as water moves into the cell and the cell membrane tries to expand, it runs into the box. It's like trying to blow up a balloon in a box. You can blow up the balloon until it fills the box totally, but unless you're strong enough to blow the air into the balloon to burst the box, which you're not, you're done. You're as full as you can go. And that's what a plant cell is like. As water moves into the plant cell, the plasma membrane expands until the box restricts its expansion, and then it stops expanding. And now it's in a turgid or normal state. So this is your plant, your house plant that's healthy, and then when you look at it and it's low on water, it's flaccid. Those leaves start to droop down because they're not rigid anymore. They're not turgid. Okay? So this is the source of that those plants going flaccid on you. Now, if we go to the other end, to the right side, the right box here, we have a hypertonic solution. And a hypertonic solution is going to be when you have more salt on the outside of the cell than inside the cell. Water wants to go where it's not, so it's going to move out of the cell, and the cell shrivels up. So in animal cells, this is a fancy word called crenulation. So the cell will shrink down like a raisin. Okay? In plant cells, the fancy word is plasmolyzed. So the plasma membrane actually pulls away from the cell wall, and these strands, and ultimately those strands will break, but those are probably where the plasmodesmata are crossing the cell wall, and as the water moves out of the living cell, right, out of the cytoplasm. So this is why salted or canned fruits and vegetables in a thick syrupy solution actually work to prevent bacterial growth. Right? We salt meat, we make jerky to preserve it, so we don't have to refrigerate it anymore. Well, what's the mechanism there? This is the mechanism. If I have a piece of beef that is heavily salted, we've drawn all the water out of it, it's dry, and as bacteria or fungal spores land on it and try to grow, all the water gets sucked out of them and they die. So the meat then is preserved much, much longer because of this hypertonic effect. All right. So this is going to take some time for you to think about. I want you to think about it in practices. Isotonic, iso means the same salt solution. Hypotonic means there's more water outside the cell. And hypertonic means there's more salt outside the cell. There's more water in the cell. And then the result is either turgid for plant cells if you're hypotonic, or plasmolyzed if you're hypertonic. Right? So practice this stuff, and we will see how that affects biology in many, many instances.